And thank you everyone for joining our webinar today about MobiuSCOPE, our new single-cell full-length RNA sequencing product that's meant to give you much more insights compared to the standard single-cell 3-end RNA or 5-end RNA sequencing solutions. And yeah, since most of you have already had quite some experience with single-cell sequencing, I would probably just skip the whole background why single-cell sequencing or single-cell omics is so important in revolutionizing our understanding of biology of disease and also in potentially serving as the future diagnostic tools and also in further promoting the new therapeutic strategies. And but here just maybe a little bit background to remind all of us why with the standard single-cell RNA sequencing, we could already get the RNA quantity information from each single cell. So we know how RNA is expressed differently in those different single cells, in those different cell types or cell subtypes. And with the quantity information, it's of course very important. We can then annotate cell types. We can then discover new cellular status, new subtypes, and we can compare their pathway activations, depressions, upregulation of genes, and downregulation of genes. Those are already very useful information, as we all know. But on the other side, if we only do like either 3' RNA sequencing or 5' RNA sequencing and only have the quantity of the RNA at single cell level, we actually miss a quite big part of the whole picture. As shown in the left panel of this slide, normally our mRNA comes from the joining of different exons of the gene. And those exons could be combined in different way to make different splicing isoforms. And as you can imagine, those different splicing isoforms, although they come from the same gene, but through different combination, you actually have real different mRNA sequences. And with those, you also have different proteins. And those different proteins would no doubt have different functions. And although they all eventually come from the same gene. And with the typical 3-end RNA sequencing method at single cell level, we actually miss this part of the picture, and we would take all those splicing isoforms as long as they have the same 3' end or 5' end, we would take them as the same, while they are functionally probably very different. And of course, there's no need for me to say the importance of understanding splicing isoforms in different health and disease conditions, because there have already been decades of study showing the isoform splicing isoforms functions serving as biomarkers, for example, in certain diseases, and also as regulators in the developmental or other essential biological processes. And besides splicing isoforms, there is another phenomenon in the cells, especially in cancer cells, that's called fusion. So this happens when a cell or the chromosome of cells would get somehow unstable, and different pieces from different genes now, which shouldn't be together to start with, would now join each other And from a totally new RNA that doesn't really exist in nature or in a normal cell. And this unexpected joining of pieces from different genes could, of course, also generate a new protein that can have unexpected function. And if things go wrong, this kind of fusion genes could really lead to diseases like cancer, especially in blood cancer. Actually, you probably heard about the Philadelphia chromosome. This is actually a quite commonly seen phenomenon. And with the standard single-end, 3' end RNA sequencing at single cell level, the fusion genes normally cannot be really detected so well either. So to be able to detect splicing isoforms as well as fusion genes in a more comprehensive, more accurate, and conclusive way, to single cell RNA sequencing is very important, but if we could also get the full length information, that would also open up a new whole window that would lead us to new insights, new knowledge, and better understanding of the cells as well as their functions. So to address this need, we developed our MobiuSCOPE full-length single-cell RNA sequencing product. And basically, the name comes from Mobius band, and which you probably have heard of. This is a mathematic concept that if you have a band and if you join the both ends together, you can form the Mobius band that will be running more or less endless from the beginning to the end in circles. And this way, as you can see here, normally for single-cell RNA sequencing, the trick is to label each individual RNA. First, to label all the RNA from each individual cell with a unique cell barcode. And after that, to label each individual RNA in this same cell with a unique UMI. So the UMI and cell barcode will be like the personal ID for each transcript at single cell level. Normally, this unique personal ID to the transcript is only next to the 3' end of the mRNA. But in this case, if we could really band together and join both 5' ends and 3' ends of the cDNA together, we will bring the 5' region and 3' region close to this personal ID of cell barcode and UMI that we can use really to identify each individual transcript at single cell level. And even with short read sequencing, because although we can always sequence full-length cDNA at single cell level, also with long read sequencing, and the long read sequencing platforms such as Oxford Nanopore and PacBio, they are of course very powerful, but on the other side, they still, the current technologies of long read sequencing is still a little bit lacking the sequencing depth as well as accuracy, as well as the cost effectiveness that are so achievable already with the short read sequencing. And here again is the technical principle. It all started with a typical single cell RNA sequencing workflow where the cell barcoding beads with poly T and will capture the poly A tail of the mRNA. And the cDNA synthesis will then start to generate full-length cDNA. And after that, we can do this circularization to bring the 5' region also close to the 3' cell barcode plus UMI, this personal ID of the transcript. And then we can generate 5-ant library by break down this circle. I will then probably show a little bit more details. This probably show the technical details a little bit better, because for the MobiuSCOPE technology, we make 2 libraries. One is your typical 3' single-cell RNA sequencing library. And in this case, you have the full-length cDNA. You don't need to do any circularization. You just do the standard cDNA fragmentation and then sequencing adapt ligation to generate sequencing libraries. And in this case, and in this library pool, the 3' region fragments will be close to the personal ID of the each transcript, the cell barcode and UMI. And then we also need a second 5' library where this circularization would be very helpful. So after the circularization, we have a reverse PCR to open up the circle again. But with the opening up of the this circle, now the 5' sequences would be quite close to the personal ID of UMI and cell barcodes for the specific cDNA. And as you can see here on the right part, then we do the normal random fragmentation of the 5' cDNA, and we do adapter ligation. And in this pool of sequencing adapters, the 5' regions are now close to the same personal ID of the same cDNA, so that if we combine both 3' and 5' libraries, we now have a mixture of fragments, all of those fragments coming from the same unique transcript from the same single cell. They will have this unique UMI plus cell barcode ID. But for the 3' fragments, we get all the reads that close to the 3' of 3' end of the mRNA. And in another part of the 5' library, we have the reads or sequences closer to the 5' end of the mRNA. And now we can collect all of the reads with the same UMI plus cell barcode together. We know they come from the originally the same transcript. And using a unique algorithm we developed, we can do assembly and piece them together to generate full-length RNA information by using simple standard paired and 150 short-read sequencing. And the workflow is quite straightforward and quite easy. The first part from single-cell partition using our microwell-based microfluidics chip, either manually or on our automation platform, as well as the mRNA capture on the chip, and then collecting all the cell barcoding bits, now bound the cellular mRNA and RT outside of the chip. So those are quite standard as in any single-cell RNA sequencing workflow that we have. And after that, RT, there's cDNA amplification. And then amplified cDNA can be generated to a sequencing library either directly or go through circularization and PCR, and then another library prep to generate the 5' library. So the whole process will take about 9.5 hours. It's a little bit longer than a typical workday. But on the other side, we do have quite some safe stop point in the whole workflow, so that you can relatively easily plan this workflow either in a stretched workday or into 1.5 days. And also with this method, you can easily get single-cell full-length RNA sequencing information from, with our standard chip up to 20,000 cells at once, with our high-density chip up to 30,000 cells all at once. So it's quite scalable and also quite straightforward workflow. And here I'm also going to show you some examples that we have generated in using this technology to get more insights from each and every single cell. And here is one example where we combined the mouse spleen cells and human PBMCs from both spleen and PBMC. Those are mostly immune cells. As you might know, they have relatively less RNA to start with, so they have relatively low RNA contents. And in this case, we sequenced just maybe as one note for MobiuSCOPE. We recommend to sequence at a depth of about 100,000 reads per cell to generate enough fragments so that we can do an accurate and comprehensive assembly to get the full-length information. So in this case, as you can see here, we could detect altogether about 6,000 UMIs per cell in medium and 2,273 genes per cell. And we have, this enables deeper detection of RNA by combining both 3' and 5' ends of libraries. And I would also like to point to your, bring your attention to the middle panel, where if you look at the percentage of those reads that are assigned to exon region, you can see, probably compared to some other standard methods, in the MobiuSCOPE methods, we do have the advantage that a high percentage of the reads, they are accurately mapped to the exon regions, meaning that we are capturing the real mRNA reads instead of maybe some aberrant intra or intergenic priming products. And because we really do need to assemble the short reads of those mRNA together to piece together like full-length information, this kind of accurate capture of the real mRNA reads is quite essential to start with. And here is the gene body coverage of the MobiuSCOPE technology. So with, as you can see, in the middle on the right part, with either 3' or 5' methods, you do see this kind of bias either towards 3' end or 5' end. With MobiuSCOPE methods, we have a relatively even coverage across different positions of the gene body. And this gives us a really more comprehensive information for the whole gene. And this question has always has been very often asked, because as we know, the medium size of human transcripts is around 2-point-something kb in a typical human cells. And of course, we would also like to know whether the MobiuSCOPE method can also have even or good coverage of the different positions of a gene regardless of the length of the transcript. And we grouped the genes based on their typical length. And as you can see here, for the smaller transcript, less than 2,220, we have good even coverage across different positions on the gene body, and same good coverage between 2,220 and 8,550. And we start to see a little bit more enrichment on the 5' end with much longer transcripts, like once they are longer than 8,550 or 28,730. But still, in general, even if with those super long transcripts, we still get relatively good coverage across like the whole gene body. It's not that we just suddenly stop to see certain positions on those genes. And yeah. And here, of course, if we do have full-length coverage with the MobiuSCOPE method, we first, with the deeper sequencing and also with sequencing from both 3' and 5' end, we avoid losing essential information. So we could detect also more genes per cell, as well as especially more splicing junctions per cell. And as you can see here from this table, we could detect more than 40,000 splicing junctions in the sample with the full-length MobiuSCOPE method compared to the typical 3' single-cell RNA sequencing method. And here is a more comprehensive table comparing the number of genes, number of UIMIs detected, as well as number of splice junctions detected with the MobiuSCOPE method compared to 3' and 5' methods. And again, we see significant improvement of UIMI, of gene, as well as splice junction detected with the more comprehensive MobiuSCOPE method. And another example to show that we do have new insights that are probably missed with the single-end single-cell RNA sequencing methods is to look at the percentage of overlapped genes here. So here is a 3' typical single-cell 3' RNA sequencing library versus MobiuSCOPE. And as you can see here, we do have overlap, and also with the increase of sequencing depth, if you look at the left panel, we do see like at the first phase, a linear increase of the percentage of reads or genes that overlap between those two methods. But at some point, this starts to get into a plateau, meaning that even with the increase in sequencing depth, at some point, we don't detect, we don't get to 100% overlap of the genes detected with the typical classic 3' RNA sequencing method versus MobiuSCOPE. We do have genes that we capture with the MobiuSCOPE method that probably have been missed with the typical single-end method. And here again, is to compare the overlap and also number of splice junctions detected with the MobiuSCOPE method versus the RNA typical 3' single-cell RNA sequencing method. As you can see here, a significant amount of new splice junctions here, almost 40,000 are detected only with the MobiuSCOPE method. And again, here is 3', 5', and the MobiuSCOPE full-length method compared. And again, quite significant amount. Number of splice junctions is detected only with the MobiuSCOPE full-length method. And yeah, and here is to show that with MobiuSCOPE method, we detect also different types of splice junctions. And from all 4 typical types of splice junctions, we detect more of them with the MobiuSCOPE method. And okay, so for the first part of the examples, we used mouse spleen cells plus human PBMC as a test system. But here we would also like to ask the question about the fusion gene. As you probably remember, other than the splicing isoforms, the fusion genes are also quite important phenomenon, especially in certain cancers. So we picked 2 cell lines, NB4 and K562, with known, first known SNPs and known mutations. Second, also known fusion genes. Because with the full-length detection of the RNA at single cell level, it also has the MobiuSCOPE also has the advantage that we can detect relatively easily expressed mutations or SNPs, no matter where they are, which position they are in the RNA. So first, just look at the sequencing matrix. As you can see here, again, with 100,000 reads per cell, we could detect from those cell lines more than 23,000 UMIs per cell, medium, and almost 5,000 genes per cell. And again, more than 90% of the reads are assigned to exon regions, ensuring that we would be able to piece them together in a more accurate way when we assemble all the reads coming from the same cDNA together. And yeah, and here this is an example showing that those 2 cell lines, they have known SNPs. And of course, not all those known SNPs at DNA level will be expressed. But what we wanted to show here is independent of where the SNP position is in a gene, whether it's small 5' or whether it's small 3'. With MobiuSCOPE methods, if they are expressed, we could detect them. So this would also make detection of expressed, for example, cancer-related mutations like driver mutations, or if we want to study allele-specific expression and to do like EQTL type of analysis, this makes MobiuSCOPE also a quite powerful tool to do this kind of studies. And of course, how do we do regarding fusion gene detection? And in this example, actually with those 2 cell lines, we know that they have known either PML, RARA fusion of the classic BCR-ABL fusion. And again, with either 5' method or 3' method, we probably would miss one or another, or we would call one or another probably incorrectly. And but with the MobiuSCOPE method, once we have the full-length information, we could also detect those known fusions in a more accurate way. And here is also a real-life sample data where we took PBMC from a specific patient. And then with the MobiuSCOPE method, we could detect a novel fusion that were not easily detectable with either 5' or 3' methods. As shown here, this fusion gene was detected in those red labeled cell populations. It's quite enriched in in two small cell clusters with unique phenotypes. And also by deeper look, we could identify the fusion as HLA-C LRRC74A fusion, which was later also confirmed with other independent methods. And yeah, so through the power of MobiuSCOPE being able to generate full-length single-cell RNA information, also at large scale, from tens of thousands of cells together, it really enables also the discovery of novel fusions and novel splicing isoforms in a very effective way. And here again, this plot also shows that in K562 cell line, we could detect those alternative splicing sites, no matter where they are regarding position on a gene. Okay. And yeah, so I talked about the application of MobiuSCOPE in discovering new splicing isoforms, in discovering or identifying fusion genes, and also in discovering or identification of the expressed SNPs and expressed mutations, no matter which at which position on a gene they are. So those are all potential applications of this method, especially probably also for drug discovery and clinical related applications, as you can imagine. But there are also organisms that are probably not so well studied. So we call them normally non-model systems or non-model organisms. They don't have a very good and complete reference genome as human or mouse do. And for the annotation of those genetic data from those organisms, if we do single-cell sequencing, actually the dropout rate, which is typical for any single-cell sequencing method, could be even more severe issue for those non-model organisms compared to organisms like human or mouse, which already have quite good reference genomes. And so in this case, actually, we used some published methods to calculate the percentage of dropout of genes with our MobiuSCOPE method compared to either 3' or 5' single-cell RNA sequencing methods. And to do the calculation, we actually used zebrafish, which is an organism with a good reference genome, because we need that for the calculation. And as you can see here, the MobiuSCOPE method, because we cover more, and we also cover from different positions of each gene, we do have significantly less dropout rate compared to either 3' or 5' based methods. This was also calculated using different dropout rate calculation algorithms. And then this would really be helpful then if we would go ahead and study like a non-model organism, such as in this case, hedgehog. And with the single-cell sequencing data here, with the reduced dropout rate, we could more effectively construct the transcript from the 5' to 3' ends, and also including the UTRs on both ends. And as you can see here from the plots here, and this would enable us to more accurately annotate all the reads to the right genes. And yeah, so that's those are like some examples of potential applications of the full-length MobiuSCOPE single-cell RNA sequencing solution. And I'm sure also, with researchers in different fields and also in with different research questions, you might also have your own ideas of how to apply this new technology to apply the further insights with your own study. And I'm really also very curious about which other applications can also benefit from the single-cell full-length RNA insights that can be generated with our MobiuSCOPE product that we probably, as developers of this technology, are not even aware at the moment. And I would also like to mention that SingleRun is a provider of one-stop single-cell sequencing workflow, starting from tissue preservation and then dissociation to generate single-cell suspension that can be used either on our manual chip or automated platform, Neo or Tensor for different throughput. So the MobiuSCOPE product can also be easily compatible with our existing automated or manual manual kits. And then, like for example, we do have the manual version of the MobiuSCOPE kit that you can start easily with only a P200 pipette without any dedicated automation instrument. But if you would like to do it in a more automated, standardized way and save labor, it's also perfectly compatible with our existing single-cell library prep automation platforms. And I would like to stop here, and thank you very much for your attention. And I'm also very happy now to take any questions you might have, and maybe we already got some questions from the audience. Yeah.
Thank you, Dr. Nan, for a great presentation, and thank you for the participants who. I can see some questions that are coming up in the Q&A session. So we can now start with the Q&A session. How much sequencing is required for a MobiuSCOPE? One question's been asked.
Yeah, because we would like to have as many as possible unique fragments from each individual transcript, so that we can do a more accurate and comprehensive and complete assembly to piece them together to get the full-length information. So we normally recommend 100,000 reads per cell for PE 150 for the sequencing. But of course, if at this stage, if the saturation is still not very high, if you would like to sequence deeper, it would definitely also help the assembly and also help to generate even more useful information. And another possibility is there's also possibility even with short read sequencing nowadays to sequence a little bit longer than P150. So if this could be extended, that could also help assembly. But the typical recommendation is P150 and 100,000 reads per cell.
Perfect. Thank you very much. The next question would be, what species can MobiuSCOPE work with?
Yeah, this is a very good question. And we actually don't really discriminate against any species, and because the technology itself only relies on the poly-A tail of mRNA from any eukaryotic cell. And we need our oligo-DT on our cell barcoding beads will capture the poly-A parts of any mRNA. No matter which species it is. It's just like once we have those RNA, we can, on the data analysis part, of course, we will need to map it to the species where the our cells come from. But yeah, the technology itself can be used for anything that would have a poly-A positive mRNA.
Mm-hmm. I guess that's one of the main advantages of this technology as well.
Mm-hmm. Exactly, yeah, and especially also for the because of the full-length information, it would be even more helpful for the non-model organism in accurate annotations.
The next question, I guess we covered a little bit, but I'm just wondering if you want to add in more. In which cases should we use MobiuSCOPE instead of a regular RNA sequencing?
Yeah. If you ask me, I would just say, like, I was thinking about this question. Of course, we can distinguish it. And there is also always a value of just knowing the quantity of mRNA in each single cell. But on the other side, I was just thinking about this question myself. Sometimes if you, for example, we started with black and white TV, for example. This is probably too old that some of you probably don't know about that. Let's start again. You started with this typical non-smart mobile phone. And then once you have the possibility of a smartphone that can do everything that this Nokia phone can do, plus much more, the question is, with more or less the same price, then the question is whether you would really still need to have this lower setting type of phone that can only generate part of the information or functions. So basically, MobiuSCOPE can do everything that a typical single-cell RNA sequencing kit can do. Like it can also generate the quantity information of RNA at single cell level to give you annotation of the cell types, to give you pathway information, gene expression information. But in addition to all those, with really minimal addition of the costs, and also more or less very similar workflow, you get much more information, such as you see the mutations and SNPs expressed across different positions of the genes in the cells. You also get to see the splicing isoforms. You also get to see potential fusions. And yeah, I would say, of course, if you have a very specific question regarding mutation, splice, and fusion, or non-model organism, MobiuSCOPE can be applied for those questions, while the typical single-cell RNA sequencing kit cannot. But on the other side, maybe even for your typical single-cell RNA sequencing projects, you can also consider whether it might be further improved or further, if you could also get those additional information that was previously not possible to generate.
Thank you, Dr. Ren. We have one more last question. If the mRNA is too long, middle part can be missed due to the lack of UMI site. Am I correct? Then what is the maximum length from the end?
Yeah, Because like when we generate a sequencing library, actually you always see this a little bit like the Gauss distribution. It's never like one very distinct size of the library. You have like maybe the medium size of 500 something, but you do have a distribution. Meaning that from other sequencing libraries, we actually get pieces that are of different distance from either 3' or 5' ends. That's how we could also piece together the information of the RNA at different positions. I would like to get back to this slide and show you that even with very long transcripts for more than 28,000, which is actually already quite extreme situation. If you look at like the middle part of the gene, which should be like 50, the x-axis 50 position, you can still detect them. And it's at the 0.8x coverage compared to the average coverage, but you can still detect them. So yeah, with this kind of random fragmentation and also assembling methods, we could also detect, even with long transcripts, the middle part of the gene, and albeit at lower coverage, at least in a consolidated way. Maybe for each individual gene, I think we need to analyze them individually.
Thank you. Are there any other questions? Or Dr. Nan, is there any additional comments that you would like to share with us?
So I thank everyone for taking the time to join our webinar, and I suppose also for some of you it's still probably still a little bit early during the day. So thank you so much, and also thank you for all the nice, very exciting and questions. And if you do have other questions or comments, please feel free also to contact us and let us know. And we can also answer you directly. And here is our contact information. And and if you have any other very exciting and novel application in your mind, and let us know, and we probably can also try something out with our MobiuSCOPE together. We already have a couple early access customers in Europe and also in the US that are doing really exciting things that we didn't even think about ourselves while developing these products. And I'm also looking forward to even more exciting ideas on how to use it from the researchers and scientists.
Thank you very much, Dr. Nan, for excellent presentation and insights. And thank you all for joining us and for your great questions in the Q&A session. We appreciate your time, and we look forward to seeing you at future events. Have a great day or evening.